Hello and welcome to your daily dose of science. In this video, we'll be discussing oncogenes and the role that they play in cancer. But first, let's refresh our knowledge. What is cancer? Well, it's a disease resulting from uncontrolled cell division in the body. Groups of these abnormal cell growths are called tumors. It's important to note that not all tumors are cancerous, but those that are, called malignant tumors, spread cancerous cells to other parts of the body, invading and destroying tissues, and that process is called metastasis. Now, in a healthy person, cells divide in a controlled manner. Mechanisms called cell cycle checkpoints moderate this process by ensuring the cell has completed all the necessary steps in one phase before moving on to the next. Cells communicate with each other through signaling pathways, and mitogenic signaling, in particular, promotes mitosis, and as a result, activates many genes that promote cell growth and division. This is why genetics play a huge role in cancer development, apart from the fact that several types of cancers are hereditary. But more specifically, changes in DNA, called mutations, called cancer by damaging DNA. Mutations can deactivate the genes critical for cell function and can also activate certain genes that they're always turned on. In a normal cell, essential genes called proto-oncogenes encode proteins that enable cell division and movement, but they're only supposed to be turned on when they need to be. But what happens when these genes are turned on all the time, even when it's unnecessary? If this were so, the cell wouldn't be able to stop moving and growing, and as a result, can't die. This is the result of a mutation in the proto-oncogene, and from this point on, it's then called just an oncogene. Oncogenes contribute to uncontrolled cell division, and as a result, promote cancer development. Fun fact, the words oncogene and oncology, which is the study of cancer, have the same root word onkos, which means bulk or mass in Greek. So what activates oncogenes? There are three mechanisms by which they're activated. The first kind is through a gain-of-function mutation, which happens when a single nucleotide base pair is added, deleted, or changed. This causes the protein product to always be on. The RAS subfamily is a common example, found in pancreatic, lung, and colon cancer cases. Another mechanism is called gene amplification, where the number of gene copies within the cells increases to the point where there's too much protein being expressed. HER2 is a common oncogene activated by this, found in breast cancer. And the third type is known as a chromosomal rearrangement, which turns proto-oncogenes into oncogenes when pieces of DNA are translocated from one chromosome to another. This also results in too much protein product or the wrong product being expressed in the wrong cell. The hero to the oncogene villain is the tumor suppressor gene. These soldiers have the job of cancer protection when they're present, and when they're absent, it creates opportunities for cancerous tumors to develop. Some examples of tumor suppressor genes are cell cycle regulators, apoptosis promoters, which encourage cell death, DNA repair enzymes, and transcription factors. P53 is an infamous transcription factor gene, and it turns on DNA repair enzymes by blocking proliferation and enabling DNA repair completion before synthesis can happen. In other words, without a fix, the cell dies. To sum up, oncogenes are essentially defective proto-oncogenes, malfunctioning in their job of promoting cell division and growth. They are activated by types of mutations that enable them to be turned on all the time, causing protein overexpression or misexpression. And as a result, the cell can't stop dividing, nor can it die, leading to tumor formation and then eventually cancer. The opposite type of gene is the tumor suppressor gene, and it helps in cancer prevention, but without it, there is a risk of uncontrolled cellular activity. Thank you for listening.
Created using Powtoon.